No matter who you are, you've been affected in some way by aging. Perhaps it's altered someone you know, weakening their body and dulling their mind. These changes will come for us too one day, and there's nothing anyone can do to stop them. Or is there? A new study by Professor David Sinclair of Harvard and his colleagues has proposed and supported the information theory of aging. This model allows for aging's reversal, which was also recorded, paving the way to reverse aging in humans. But to understand the information theory of aging, we must understand epigenetics. Most people are familiar with genetics, the DNA that determines our traits. Epigenetics is the less famous second layer of control. It regulates which genes are expressed. This is important because each organism's cells are almost genetically identical, and epigenetics is what makes the correct traits appear at the right place and time. To test the theory, Sinclair and his colleagues used a two-part treatment, each made up of DNA coding for proteins, Cre-Er, which triggered I-papi, and I-papi, which then created double-stranded breaks in mice's DNA. Together, they form a system called ICE, inductible changes to the epigenome. Control mice received either Cre-Er or I-papi, while test mice were treated with ICE, which made it very easy to repair breaks, thus avoiding mutations. However, repairing the breaks changed the epigenome as predicted. In a month, the ICE mice prematurely aged to middle age, and in 10 to old age. How did this happen? I present the Waddington landscape, a metaphor for cell differentiation. All of our cells started out the same, but due to the epigenetic landscape, diversified, with the combination of each type's genes and their interaction being a valley. The landscape was smoothed as reorganization increased contact with DNA outside each valley, and communication inside decreased. Enhancers, DNA which enhanced the expression of genes, saw the same pattern. The smoothing was reversed by three proteins called Yamanaka factors, OCT4, SOX2, and KIL4. The genetic codes were engineered into the small harmless adeno-associated virus, which then delivered the DNA into the ICE group cells. The cells began producing the factors, which instructed the cells to begin undifferentiating, undoing the epigenetic changes. They stopped before losing their identity, but restored the Waddington landscape and de-aged the mice by up to 57%. After 5 weeks of this treatment, analyses could no longer distinguish them from the control. This is extremely exciting because it paves the way for a near future where human aging can be reversed and no one has to grow old. Imagine that world. And when it arrives, we can thank the Sinclair Lab.